No, I don't have your exam. You'll get it on Tuesday. Today we're going to go over two domains, bacteria. That's okay, I can talk really fast. So we're going to go over bacteria and Archaea. But one of the things about biology is you can't always predict what you're going to find or what you're going to see. So last night I put my dogs out and a praying mantis got into my house. So the kids and I are trying to get the praying mantis. Of course I tell my youngest one, go get your bug jar, and he says I have toads in it. So, hence I find out I now have a toad in my house that I did not know, but I brought it in. So unfortunately I am very sorry it's in their, those little candy things. Jars. I tried to scrape it off, but take a look. Do not let it out, okay? And the reason why is because if I do not bring it home to my son, instead of grading your exam this weekend, I will be searching all over to find a praying mantis. I promised I would bring it home to him. But go ahead and take a look. It's not very often. This is most likely a female. Females tend to be larger. Okay, this is an insect. They get their name because when you look at them, they have these upper arms that go like this. They look like they're praying. They're still hunters, so they stay there. There are different kinds. We have some others that are native, that are more of a brown color. They're more in the deserty area. This one's green, so you really have to look for her. They have some interesting mating behaviors. For example, the female bites off the male's head while they're copulating. She eats his head, so hence he dies. Okay? Yep, that's typically what happens. The female reaches around and eats him. Very likely a female, kind of a big one. I don't know how to sex praying mantises, but kind of a cool animal. Okay? So it's a predator. It eats insects. And it's very good at eating insects and catching them. Please be very careful with it, though. Yeah, don't hit it. Okay, I got to get through a lecture I had intended on doing in 2 days in one. So there's a lot of stuff that's already written down, thank goodness. All right. So we're doing chapter 27. This will be on exam 2. Okay, there are three sections on exam 2. This one, then the protists, which are a lot, and then fungi. Okay. All right, so most often, bacteria and Archaea, most are unicellular. Uni meaning one cell. They have about one cell. Some of them not. Their size, they're very small. Go anywhere from 0.5 micrometers to maybe about 5 micrometers. There are some that are even smaller, but typically about 0.5 to 5. Now there are different shapes that we identify. Cocci are these circular or round shapes. Okay, so they're in the purple. See how they're these rounded shapes? I call them spherical because they're 3D. When we talk about diplo, we're talking about 2 of them attached. If we use the prefix strepto, we're talking about chains, so they exist in chains. Staphylo, they exist in clusters. Bacilli are rod-shaped. That's the second picture there. Okay, see how they have this rod shape? Most of them are singular, so unicellular. Okay, but we do find them in chains. Spiral, there are different types of spiral. We can have what's called comma shape, loose coil, or kind of like a corkscrew. Spirilla tend to be kind of the loose spiral or the comma shape. Kind of a loose spiral. The one that's pictured here, this one, okay, is a spiral key. This is a corkscrew type. Tightly coiled. Okay, so those are different shapes that we see in bacteria. Cell surfaces. So things that make up the cell. Now I know we haven't gone over cell. That's really bio 6. But there are structures that make up cells. We're going to be talking about prokaryotic cells, so before the nucleus. And one of the things that we see that make up the bacterial cell will be cell walls. Now, cell walls function to protect the cell. They protect the cell not only from other organisms, but from bursting. They also make sure that the cell keeps its shape. All right. One of the main things that we see in the that makes up the cell wall or associated with that cell wall is a material called peptidoglycan. Okay? It's made up of sugars and polypeptide cross-links. Now, depending on whether or not there is an immense amount of that material or whether there's much less, actually helps us determine what type of bacteria we have. Because we use something called a gram stain. You guys did that in lab, right? Okay, so you guys should know this one. Great. Okay, so this peptidoglycan is a portion of that cell wall. Bacterial cells have them. The domain bacteria have it. The domain Archaea do not. They do not have peptidoglycan. I'm sorry?
It helps determine what?
It helps to determine what type of bacteria we have. Okay, so what I did here is I divided the two groups into gram-positive and gram-negative. And that's using a procedure called the gram stain, which you did in lab. Okay? So you guys remember what you did in lab, right? You dyed it with, you can read it, it's up here, crystal violet. Did you guys dye it with the crystal violet? Okay, I haven't done that in lab, so to explain, tell me if this is what you did. Crystal violet, then you washed it with alcohol, then you dyed it with a red dye. Yeah, that's a gram stain. Okay, so what happens is, if you have this structure here with lots and lots and lots of peptidoglycan in the cell wall, that's this clear portion here. When you stain it with the crystal violet dye, that goes into the cytoplasm. Okay? But when you go to wash it with the alcohol, you can't wash it out. The peptidoglycan prevents it from washing out. So a gram-positive cell occurs because it has a lot of peptidoglycan. And this traps the violet dye into the plasma membrane. I'm sorry, into the cytoplasm. Okay, so you get it trapped into the cytoplasm. So when you go to wash it with the alcohol, you don't wash that purple out. It can't escape. So the alcohol does not wash away the purple dye. So when you put the red dye on it, it doesn't stain. You can't see the red through the purple. So these show purple. And if you look at this little picture up here, it's in your book. See the really, really purple ones? Those are gram-positive. whereas the pink ones are gram-negative. So what happened here is in the pink, you don't have as much peptidoglycan, and you have membrane on both sides. So when you dye it with the purple, you get the purple dye in, you then are able to wash the purple away from with the alcohol, and you put the red dye on, it shows this pinkish color. That's a gram-negative. Okay? Does not have as much peptidoglycan. Okay? And that's this diagram here. Does not have as much. Here's the peptidoglycan here. But notice it has a membrane on the outside and a membrane on the inner portion of that peptidoglycan. So both outside and inside. This allows us to wash away that purple, and so the cell shows up pink. So you guys did this in lab. Okay. Any questions? Just reiterating what you did in lab, basically. All right. Some treatments, okay? Knowing what type of cell or what type of bacteria, gram-positive or gram-negative, will help us identify what type of antibiotics to use. You don't want to use a certain type. If you know a certain bacteria is gram-negative and you stain it and it comes out gram-positive, you know you don't have that type, right? So you want to be very careful. That's one of the things that they look for is which class or which group that bacteria are in to help identify what's going on. All right, let's see. Capsule. This is another structure of bacterial cells that we see. And the capsule, enlarge it here, is this green portion here. Surrounds the entire cell. It's this sticky type of material. And what it does is it allows the cell to not only stick to others like itself, but to surfaces, to other materials. So it functions to help protect the cell from dehydration, and also allows the cell stick to surfaces and other organisms. It's a polysaccharide, or sometimes even a protein. It encompasses the entire cell, so it goes around the cell. And that's that green portion here. All right, so this is another component of the cell. So any questions? I told you guys to look at eukaryotes and prokaryotes before. The cells, I told you to go back in your book and take a look at them. No? Yes, I did. It was like the first day. I said, you guys got to look at it. We're not going to go over them because I don't have time, but you should probably look at it. Okay, well, after this, you should go look at prokaryotic cells. They're much simpler than what we call eukaryotic cells. So we have prokaryotes and eukaryotes. Pro means before, U means true, karyote means actually means kernel, but it's referring to the nucleus. Eukaryotes have a nucleus. Prokaryotes do not have a nucleus. Okay. DNA, all living organisms have DNA, but not a nucleus. Okay, so both prokaryotes and eukaryotes have that DNA, but it's different, located in different ways. Right? You really should take a look at the cell, because I'm going to be going through a lot of it. And I'm just going to expect you to understand it. Okay? Yes?
What do you into the structure of the capsule?
Oh, it's either a polysaccharide or protein. Fimbriae. This is another structure that we find make up bacteria. These hair-like appendages, see these little appendages that stick out? Those are called fimbriae. And this also allows the cell to stick to substrates or other organisms. So these little hair-like appendages that stick out from the cell that allow the cell to actually stick to something. It attaches. It's for attachment. Sex pili. This is another structure that a lot of bacteria have. These are extensions to the cell that allow for exchange of DNA. They tend to be longer than the fimbriae. Okay, so they tend to be longer structures. You guys know how I test. Can you think of some questions I might ask? I've already seen one test. I figured the first test is a preview, right? It's one of those, you don't have any clue who I am, what I want. Did I miss anything on the last test? Was there anything on the Elmo sheets that I missed? No, because I took my Elmo sheets and I went, oh, I like that one, and I typed it up. Oh, I like that one, I typed it up. That's how I wrote the exam. I went from sheet to sheet to write the exam, the questions. Okay, so I did not miss anything. I might have missed one or two words because there were only so much time. If I had had the 3-hour lab to give you the exam, I wouldn't have missed a thing. Okay? But I didn't. I only had an hour and 25 minutes to give you. All right. So movement. Taxis. Taxis is directed movement toward or away from some stimulus. I'm sorry? Oh, to facilitate conjugation. I'm going to do conjugation after. That's a form of reproduction. Actually, it's a form of exchanging DNA between two bacteria. If a cell does what we call chemotaxis, it responds to a chemical. It either moves towards a chemical or away from a chemical. Okay? Phototaxis, towards or away from light. So taxis is a directed movement. They either go towards something or away from something. And if they go towards a chemical or away from a chemical, it's called chemotaxis. If they go towards or away from light, it's called phototaxis. One of the main structures that we see for movement is a flagellum. Flagella is plural. The flagella of prokaryotes is much smaller than that of eukaryotes. Actually, if you look at the flagella of eukaryotes and bacteria and Archaea, it looks like they all are analogous. They're not homologous. It looks like the flagellum itself did not come from a common ancestor. Rather, they all arose independent of each other, which is kind of neat. Okay. All right. There are 3 parts. There's the filament. There's the filament, which is this portion here. Okay. The filament swirls, okay, and it moves. And the way it does movement is it actually propels the cell. Do you have a question? Oh, yeah. How far up? Okay. You're welcome. Okay. So it propels and it rotates, and so it moves, it's able to move the cell. The way the flagellum actually increases in size, you don't have to know this, but I found it so neat, is they add parts to the end of the flagellum. So you increase by adding parts to the end and elongating the flagellum that way. The hook is this portion here, that portion right there, and that turns. So the hook turns, and the flagellum's going to rotate to propel the bacterium. The motor is actually this entire thing. It's made up of a whole bunch of proteins. Okay, so the motor is this entire portion here. It's made up of a whole bunch of proteins. No, you don't have to know the proteins, you just have to know the motor. Okay, it's this portion here. It includes a rod, which is this middle section. Now this is what's really interesting about the protein. I'm sorry, about the flagellum. This is the bottom portion. Okay, and it's a series of rings, protein rings. One of the things that creationists like to use as saying that evolution cannot happen is the flagellum. And they make a very valid point unless you listen to what they're saying and the evidence that we have at this point. You need the pieces of the flagellum to work. You can't just, and they're actually from modified pilus-like tubes. Okay, so remember we were talking about pili, sex pili, the longer attachments. They're modified to a flagellum. Well, one of the points made is how can you get, there's something like, 42 proteins. There's 42 proteins that make up this flagellum. I mean, it's not a simple little structure. It's a big thing, right? So how can you get something that is that elaborate just appear? Makes a good question, isn't it? Right? I mean, that's a good question. How can you get something that you need all the parts in order to work to just start working? That's a very good question. Well, this is the problem. One, there's about 42 proteins. Only 19 of them are essential. Every protein is duplicated. It's a modification, a mutation of some duplicated ancestral protein. They're all similar. So it works beautifully. Because what they've seen is differences in organisms on the complexity of the flagellum. And not all the proteins are needed. So all the proteins that are in the flagellum are not essential for it to work. Okay? That's why the argument that you can't have a flagellum develop over time doesn't hold. And that's because there are 42 proteins, only I think about 19, no, about 21, sorry. Only about 21 are essential. 19 of them are modified versions of some other protein. Remember when we were doing evolution, what was one of the main things that had to happen to a gene that was essential? You had to duplicate it, right? So you had to get a duplication. Now you got two of them, you can modify one of them. Because you got one that's essential. That's what they've seen. They've seen most of these are duplications of some ancestral gene. They're all too similar to each other. Okay? So evolutionarily, the flagellum is a perfect example of something we call exaptation. In exaptation, what we find is that it's a process in which structures that already are existing things that are already there are used for something different. They're modified for a new function. Okay? So we're going to actually see a lot of this during the class, exaptation. So you have existing structures, and they're modified for different functions. And they can be slowly modified, they can be minutely modified, or there can be major modifications depending on the changes. Okay. Internal organization. Key characteristic of living. Do you guys know life's processes? Okay. Not on the exam, but essential to biology. Let's do some of the life's processes, things that life has. Organization. Complexity. I'm not going to do all of them. Metabolism, which is basically chemistry. Form chemistry, they get energy, they make things. Homeostasis, they have to stay within a range. If you go outside that range, they either have to get back in the range or something happens. As groups, they evolve. Which is basically change over time. Respond to stimulus. They respond to stimuli. But one of the main is DNA, deoxyribonucleic acid. Okay, living organisms have DNA. In the case of prokaryotic cells, that DNA is not found within an organelle called the nucleus. In eukaryotic cells, it is found within an organelle of the cell called the nucleus. But in prokaryotic cells, it is not. It is found in a region we call a nucleoid region. So we have a circular DNA, or prokaryotes have a circular DNA, found in a nucleoid region. And if you look in your book, this didn't come out very well, but if you look in your book, this will show you one very large chromosome. It folds around it. Okay? In addition to that, don't talk in here. In addition to that, they have separate parts of DNA called plasmids. These are small regions of circular DNA. And they replicate on their own, which means they make a copy on their own. Okay? So no nucleus, no separate organelle, membrane-bound organelle. Okay? So there's nothing separate holding them, but the chromosome, the DNA, is in a circular pattern in a generalized region called the nucleoid. Okay, so any questions? Okay. Reproduction and adaptation. Bacterial cells go through a process we call binary fission. It's not mitosis, it's not meiosis. It's got a specialized name. We call it binary fission. It's when the circular DNA replicates itself And then the cell elongates and pinches off into two separate cells. The type of reproduction, and it's very fast. It can occur, a single cell can divide in 20 minutes. It can divide in a couple hours, but it can divide in 20 minutes, which means you can double a population of bacteria every 20 minutes. That's why a bacterial infection can be so dangerous, right? Because if you got one, then in 20 minutes you got two, 20 minutes later, 4, so it goes up exponentially. They divide very, very quickly. And this is where a single cell gives rise to 2 cells. The single cell's gone. It's not mitosis. Okay, it's a specialized binary fission. All right, there are 3 key features to the reproduction and adaptation ability of prokaryotes. One is they're small, very small. They reproduce quickly, so very short germination time. They reproduce, they have a very short lifespan. And they use binary fission, which is a fast type of reproduction. So short germination time, relatively a low, short lifespan, and they reproduce very quickly. All 3 of those characteristics allow for very rapid, very fast mutation. So you get a population that can change very, very quickly in response to its environment. Okay? So if you remember evolution, when we were talking about gradualism, we're saying, well, over time, things can change, right? The environment can act on genes, and over time, long periods of time for gradualism, you get change in the population, or microevolution. From one generation to the next, all you have to have is a point mutation, and that's microevolution. For bacteria, even though mutation tends to be relatively rare in comparison to all the DNA that's being copied, they reproduce so quickly that mutations or changes in the DNA can accumulate very fast. In addition to that, they can withstand very harsh temperatures. Anybody know how hot the geysers are in Yosemite? I mean, they're more than boiling. No? I don't know. I was just wondering. Yeah, there are bacteria that live in there. Okay? There are bacteria that live at the bottom of the ocean floor next to vents. So they live in some very harsh environments. Okay? One of the things that prokaryotes are capable of doing is producing something we call an endospore. An endospore is basically a copy of DNA that's encased in a cell but not viable. So the cell actually dies and the DNA is protected. And when conditions are favorable, the endospores are released, and then they can produce new bacterial cells. So they have all the characteristics necessary for very fast evolution, very fast changes in populations. No, we'll see endospores in other places too. We'll see spores in other places. Yeah. It's usually replicated by a cell when conditions are becoming unfavorable, and then put into this protective dehydrated cell, so basically it doesn't have any water, and it's not active. And then when conditions become favorable, it's released and replicated into a new cell. Kind of, yeah. Yeah, in a way.
So natural selection doesn't really work
Oh, yeah, it works.
because they preserve their cells.
Some do. Not all. Not all produce endospores, but some do. We're really good at artificial selection on bacteria. You guys been hearing of superbugs? Where's the worst place to go for disease? Where can you get the most prolific, dangerous disease? Usually the hospital. Yeah. Why? Because you're go to the hospital and you use antibiotics. I'm not saying don't use antibiotics, but you have to use antibiotics when you have bacterial infection, antiseptic, you're constantly cleaning, everything needs to be sterile. I mean, it makes sense that everything's sterile. Problem is, there are some individuals in a population that have the resistance gene, which means, do you guys remember when I was talking about DDT? And I said, well, the only way you're going to kill an insect with DDT is crush it with a can? Yeah, well, it's the same idea. The only way you're going to kill some of these bacteria is we've now made populations that are resistant to the antibiotics we have, and so we have to produce different antibiotics. And that's what's constantly happening. We kill the individuals that are not resistant, but there's just one or two. Well, look at how fast they replicate. If you've got just that one or two, then you're going to get a new population. And so that's why you're constantly having this mixture of medicines. Okay? So sometimes you'll get an infection in a hospital and it can be very, very dangerous. Okay? Because you're killing off the ones that aren't resistant. Then you've got to figure out what the ones that are resistant are not resistant to. Okay?
So when somebody from the hospital gets like an infection,
Yeah,
they're
the
getting
staph
the
infection.
like they're getting the medication. So would that person have a relapse and have the strong?
Not necessarily. Not necessarily. Depends on the population of bacteria. And generally what happens is you don't just go home. If you have a serious problem, they don't just say, here, no, I can't say that. What do they tell you? If it doesn't work in 14 days, come back? That's because if it didn't work in 14 days, you probably have another strain. You go back, you get another antibiotic. But that's why, in the 70s, it was like antibiotic, oh, just kill them all. Now we're realizing you have to be a little careful of that. They're not supposed to just hand out antibiotics. They're supposed to figure out what you've got before you just start shoving antibiotics down people's throats. That's one of the reasons.
What is the function of endospores? What kind of function?
Endospores is to protect the DNA information. So when it's not favorable, when those cells can't grow, and we'll actually see this throughout the kingdoms that they are mechanisms that organisms have, so that when conditions are not favorable and the population is dying, you produce these structures like preserving. You produce these structures that can withstand these unfavorable conditions. And then when you get these favorable conditions, they can grow again, and you get a new population. Okay? So they're there to go to another population. Yes?
If we can stand harsh conditions, how does it make sense to sterilize the infection?
Not all bacteria can do that, and not all Archaea. It's mostly Archaea that withstand severely harsh conditions. Now, I'm just talking about remember, in microevolution, not all individuals are equally alike. That's why I did evolution first. There's variability within the population. If you have an organism, an individual that has one of those variabilities, and your medicine doesn't work on it, then that individual, by binary fission, when it comes to prokaryotes, are going to be able to reproduce much faster. Okay? But that does not necessarily mean that just because you have a population of individuals that you have one that would be resistant. All right, 3 factors that give rise to genetic diversity. Genetic diversity leads to what? Or can lead to? What do you need genetic diversity for? Yeah. Without genetic diversity, you're not going to get evolution. Okay. So one of the factors is they reproduce very quickly. And this is by binary fission. Mutation. Although mutation generally is considered rare, it can increase in a population that is reproducing very fast. If you have a short generation time and you're constantly making new individuals, you are getting an accumulation of quite a few mutations. And so that increases genetic diversity. And the third is what we call genetic recombination. When we talk about a cell that's a recombinant, we're talking about a cell that has DNA from more than one type of individual. So it's a recombinant. Now there are three types that we talk about: transformation, transduction, and conjugation. And this is where that facilitate conjugation will be. So all three of those are able to bring DNA together from different individuals, and they do so differently. So are there any questions so far? All right, transformation. That's one type. In transformation, a prokaryotic cell is able to take DNA from its surroundings and incorporate it in its own DNA. It usually just takes it from the environment. The way they typically incorporate is they exchange homologous genes. That's transformation. Transduction, we see something we call a bacteriophage. And this is actually a virus that infects bacteria. And then it actually will take the DNA from one bacterial cell to another bacterial cell, not on purpose. And if you look, right, so this is the donor cell, and this is the virus. And what it'll do is it causes the DNA in the donor cell to replicate its genes. And then it packages those genes in additional viruses and releases them. But every once in a while, a gene from the bacterium gets stuck in the virus. And this leaves. It bursts the cell, the bacterial, lyses the bacterial cell, and it is expelled into the environment. And then when it goes to infect another bacterial cell, instead of depositing its genetic material, instead of taking its genetic material and putting it in the new cell, it takes the genetic material that it took up from the donor cell and injects that. If it's homologous, they can exchange, just like you would in any homologous pair, you can exchange the DNA. And so now you have this cell contains DNA from another individual, called a recombinant. Okay? Virus didn't do it intentionally. It would have been an error that occurred during the infection process. So it took up some of the bacterial DNA rather than its own genetic material. Yes? I don't, I'm sorry. I would imagine if the bacterium replicates every 20 minutes, it would be about the same, but I honestly do not know the amount of time. I think it's different for each one. It would depend on the bacterium and the virus. But again, I'm not positive. Any other questions? This is one of the ways we move genetic material from cells to cells. We use viruses to move genetic material from one cell to another. I'll get that in 5 cells. All right, third type, conjugation. Okay. Conjugation, plasmids play a very important role in conjugation. Okay. In conjugation, this is where individuals exchange DNA. Okay, so that genetic material, oh, I'm sorry, it's not exchanged. That genetic material transfers. Okay, so an individual will transfer DNA or transfer the genetic material to another cell. It's a one-way process. So you go from what we call an F positive to an F negative cell. And F refers to fertility, the fertility gene. Okay. So if you look here, it's using that structure, the sex pili. Here's a cell. Here's a cell. There's the formation of a sex pilus. There's only one here. This allows one cell, if it's probably more that direction, it allows the one cell, the donor, to transfer genetic material across to the other cell. Okay? It's directional. It's only going to go in one direction. It goes from what we call an F positive cell to an F minus cell. So the cell that has the fertility gene to the cell that does not have the fertility gene. This is a sex pilus. It joins the two cells. And that's what facilitate conjugation at that point meant. Okay. When I did the sex pili and I had facilitate conjugation and I said I'd get to it, that's it. All right, so we're going to do the two types. What if the fertility gene is part of a plasmid, and what if the fertility gene is part of the actual circular DNA? So there's a few requirements. Oh, I'm glad I wrote them down. Okay, so you have to have this F factor, or the fertility genes. Okay, it's made up of quite a few genes. If a cell has that F factor, it's called F positive. And it's going to be a donor cell. If it does not have that F factor for those genes, it's going to be an acceptor. Okay, so it's F minus, and it's an acceptor. So a donor gives, an acceptor receives. Now the F factor can be a separate plasmid, okay, so that's one, or it can exist within the circular chromosome. And you can actually, bacteria can actually take the F factor and transfer it by way of conjugation, transfer it to another cell. So if you have the F factor as a plasmid in a cell, so if there's a bacterial cell that has that F factor as a plasmid, typically it's as a plasmid, and you transfer that from one cell to the next, you can cause an F minus cell to become an F positive cell. All you did is you took the F factor from one, the donor, and gave it to the F minus. Well, now that the F minus has it, it's called an F plus because it now has that fertility factor. Okay? So you can actually transfer it. Doesn't necessarily work that way if it's part of the chromosome, because generally what happens is you need to have some type of homologous transfer. So you got to exchange homologous genes, and if you don't have that, you're not going to exchange that into the chromosome. Yeah.
Is it all the way around?
No,
So how do you get F minus factor?
not F minus, you transfer the F positive. You go from an F positive factor, you take that factor as a plasmid, you put it into an F minus cell. It's not the factor that's called F minus, it's the actual cell. So if a cell does not have the F factor, it's called an F minus. You transfer it over and you get, that F minus can now become an F positive. Right? All right, so the F factor. So if you have the F factor in a separate plasmid, this is called the donor cell. Notice this one does not have the F minus in there. Okay? It's the acceptor. The F plus cell, again, the donor, the F minus cell, the acceptor. Okay. The F plus cell, the fertility factor, allows the donor to produce a mating bridge. Sex pilus. It's just biology. They got to call it something else too. Yeah, typically it's just the sex pilus, but it's called a mating bridge. That mating bridge allows the DNA to go from the F positive, which is here, up here, into the cell that didn't have the plasmid. Okay? Just replicate it. It's not like it moves. It replicates a copy, and that plasmid is able to go into this cell. Okay? Any questions? Okay. At some point, that mating bridge is going to break, usually when the plasmid gets in, and this is now an F positive cell. And that one's also an F positive cell because it didn't lose anything. It just conjugated. Took its DNA material and put it into another. Because remember, plasmids are separate DNA material. Okay, so any questions? All right, what if the F factor is part of the chromosome itself? It's a little different. What happens is we have what's called an HFR cell, high frequency recombinant. High frequency recombinant. In other words, it has the F factor and it's able to take DNA from its cell, produce a mating bridge with another cell that does not have the F factor, and take DNA material, put it into the other cell. Right? So it produces a mating bridge. It goes through replication, so it copies its DNA. Usually contains the F factor in it. Okay, so it copies its DNA. And a portion of that DNA goes through the bridge into this F minus cell. Okay, does everybody understand that? Are there any questions? You never get all the way. It doesn't make it all the way across. Something happens, the bridge breaks. It cannot, it's not going to exchange genes because it didn't have that F factor. So it exchanges the homologous portion. It can exchange the homologous portion that it got from this F positive cell, but it still stays F minus because enzymes in the F minus cell break apart the F factor, break apart the material, so it does not keep the F factor. It doesn't become an F positive cell. All right, so if it's a plasmid, you can go from an F positive cell, you're going to go from an F positive cell, which is the donor, to the F negative cell. You can convert that F negative cell to a positive because it can keep that as a plasmid. But if the F factor is part of the circular chromosome, it doesn't. The recombinant, the thing that now has DNA from a different individual, this recombinant doesn't keep the F factor. So it's not going to make a mating bridge with something else. Okay? The DNA is taken apart. All right. So any questions? Okay. No, I'll show you. It's okay. If you remember, in a prokaryotic cell, there's two places that you have genes or DNA. You have it in a circular chromosome, okay, in the nucleoid region, and then near that you have it in plasmids, which are separate segments of DNA, separate portions. Okay. In this one, it's showing you a plasmid. It's not like this one doesn't have the plasmid. It just doesn't have the F factor in a plasmid. So since that's what we're talking about, they're not going to take the money to draw it. Okay? So this one, they have the F factor in the plasmid. Okay? This, you can actually transfer the entire thing to another cell, a cell that doesn't have it, and you convert it now to an F positive cell. This one, the F factor is actually in the chromosome itself, the circular chromosome in that nucleoid region. You are not going to take the F factor and insert it into this chromosome. Okay? The F factor allows this donor cell to produce a mating bridge, but you're not going to convert this cell into an F positive cell. What you're going to do is get some of the DNA from this circular chromosome into this acceptor cell, and you're going to get crossing over. You're going to get exchange of genetic material. Okay? It's actually eaten apart. I'm sorry. Enzymes break it apart. Okay. Any other questions? Yes.
When the enzymes break it apart, do they break it down completely?
Yeah, typically you're gonna, that's material. They can actually use material. I mean, you guys know what a nucleotide is made out of. So you've got sugar, you've got bonds there, so you've got carbons, you've got hydrogens, you've got nitrogen base. So yeah, it can use that material, but it's not going to use it as the F factor. What else? Okay. There are other types of plasmids. I'm not going to go through all these. I just don't have time, but I wrote it down. Okay. Like our plasmids, it's resistance to antibiotics. So some bacteria, not all of it, have our plasmids that give them resistance to certain antibiotics. And you can actually read this. I wrote it down for you. Okay, nutrition and anabolic adaptation. All right, so some nutrition. Prokaryotes are categorized by how they obtain energy and carbon. Okay. There's two ways to obtain energy. They're phototrophs and chemotrophs. Phototrophs obtain it by light, chemotrophs by chemicals. You can obtain carbon as an autotroph or a heterotroph. Autotrophs get their carbon inorganically, typically CO2. Okay. Heterotrophs need their carbon organically, organic molecules. Okay. So you can obtain energy by using light or chemicals, chemicals being organic chemicals. You can obtain carbon by inorganic, CO2, carbon dioxide, or organic.
Actually, my paper doesn't have that portion. This. The one that starts with and carry.
Doesn't have this? Okay, write it. It's in your book.
Oh.
Or the easier way is just to take a picture of my notes when I'm done, which I will allow. So I'll share. Yeah, you guys are welcome to take pictures of these. I don't particularly care one way or the other. All right. Okay, major nutritional modes. We have the autotrophs here, produce their own food. Heterotrophs here need to consume something else. Okay? If you look through them, it goes through what they need, their energy source and their carbon source. I took this right out of your book, page, table 27.1. Okay? So you need to know photoautotrope, light and carbon dioxide, chemoautotrope, inorganic chemicals and carbon dioxide. Photoheterotrope uses light and organic compounds, and chemoheterotrope, organic compounds and organic compounds. Okay, I'll go over it real quickly. Okay, photoautotrope. Photo refers to light. Autotrope means they can use inorganic carbon to produce organic material. A chemoautotrope, chemo is referring to that you have to use inorganic molecules. Okay. But they're autotrophs, which means they can use inorganic carbon dioxide to produce organic molecules. So these are categories of organisms. Autotrophs produce their own food. Their food being they produce organic material. Heterotrophs need to consume something to get organic material. Heterotrophs, we have two types, photoheterotroph. They still are able to use light, but they have to consume something to get their carbon. Okay. Chemoheterotrophs, think us. Okay. We can't use light. We get our energy from organic material, and we get our carbon from organic material. So photoautotrope, think plant. Chemoautotrope, think human. The other two are pretty much protists. Pretty much prokaryotes. All right, role of oxygen in metabolism. So how is oxygen used? Do anybody know what obligate means? Obligate? Yeah, obligate. What is it? Have to have. It means you absolutely have to have it. Aerobes. And what do they need? Oxygen. So obligate aerobes have to have oxygen. They use oxygen for cellular respiration. Obligate, they absolutely cannot have oxygen. Obligate anaerobes have to have no oxygen. Oxygen kills them. Typically they do what we call fermentation. Aerobes have to have oxygen. They use oxygen. Anaerobes don't use oxygen to extract their energy. Facultative anaerobes, they can use oxygen or not. In other words, if oxygen's there, they'll use it. If oxygen's not there, no big deal. They don't use it.
So for the anaerobes, if they come in contact with oxygen, they'll like...
The obligate anaerobes, it'll kill them.
Yeah.
Yeah, it'll die.
So what are they usually do?
I'm sorry?
What do they usually do?
A lot of them are going to be like Archaea. They're going to be deep sea vents, places where you're not going to find oxygen. Marsh gas, so you find them in marshes. Yeah.
What does the CR stand for?
Cellular respiration. Okay, nitrogen metabolism. Prokaryotes can actually metabolize nitrogen, okay, in different ways. They convert atmospheric nitrogen to ammonia. It's called, in a process we call nitrogen fixation. We can't do that. We need that conversion in order for us to get nitrogen. Our nitrogen, we can't convert atmospheric nitrogen. We actually rely on other things to get that. Okay. So what does metabolic cooperation mean? Well, it simply means that organisms use other organisms to obtain materials that they themselves cannot obtain. the nice thing is, in the lecture, I don't have to give you a whole lot of names. You probably can get a lot of that in lab, a lot of identification and things, but there are a few you have to know. And it's just one of those you just have to know. Cyanobacterium, anabine. You need to know anabine. Okay, you just have to. They're photosynthetic, fresh water. They contain something, they form these filaments. They form these filamentous chains. Here are these cells. One of the things that happens is anabine is capable of doing what we call nitrogen fixation within specific cells. These little cells here, okay, called heterocytes. Heterocytes perform nitrogen fixation. The regular cells, the normal cells of the chain, of the filamentous chain, perform cellular respiration with oxygen. The problem is the anabine needs to separate them because the enzymes, oxygen destroys the enzymes needed for nitrogen fixation. So you've got these two different areas. You've got these typical cells that do cellular respiration, and then you've got these heterocytes that do nitrogen fixation. And you've got to separate them because the oxygen that's produced by these normal cells will actually destroy the enzymes that the heterocytes are using to fixate nitrogen. So there's a separation within the chain itself to make sure that its own enzymes don't destroy what's going on. All right. Sometimes you form colonies of biofilms. So you ever looked on the surface of water and you see this, like, I don't even know how to explain it, just called mass of material, just on the surface, kind of a glossy mass. That's a biofilm. Okay? That's formed by prokaryotes. Ocean floor, we have something called sulfur-containing bacteria. So it actually uses sulfur instead of oxygen. Gives off methane gas. It's that? Like that? Okay. All right. Any questions? Because I'm not going to finish today. Nothing? I'm not letting you go either, but I'm just not going to finish, so. No? Okay. Our favorite subject, phylogeny. Do you need to know these? Yes, you do. Sorry. Is it going to be fill in the blank? Probably not. Okay? Most likely not. I haven't written it yet. You got to remember, I don't write until after I do the lecture for what I get through. Okay? So think matching. Okay? It's more likely, especially for the prokaryotes, I would do matching for the terms. Okay? For the names of the phylogeny. What is this here? Oh, yes, thank you. Yeah, it's a clade. How about this? Terraform? How about if I do that? It's a clade. What are the domain Archaea closest to? That's closest relative? Ancestor. Came from a common ancestor. Yeah, domain eukarya. They believe that domain Archaea, is more closely related to domain eukarya than either one is to domain bacteria. A lot of it has to do with the proteins that they contain, because they have a lot of the similar proteins. Even though Archaea and bacteria structurally look similar, they're not. A lot of the molecular portion of it, they're not similar. Hence, the table. This is in your book. Do you need to memorize it? I know you're afraid to say yes or no, huh? Yeah, there are a few things you have to know. Am I going to hand you the table and say memorize it? No. I'm not going to tell you to fill out the table. So at least that's good, right? But there are a few things you need to know. You need to know their nuclear envelope. In other words, do they have a nucleus? Okay. Neither bacteria nor Archaea have a nuclear envelope, but eukarya do. Membrane-bound organelles, neither one of them have. I try to highlight them in purple. So if they're highlighted in purple, they're similar. They have the same. Peptidoglycan, look, Archaea and eukarya, neither one of them have it. It's one of the things that make them very similar. You can look, membrane lipids, bacteria and eukarya don't have branched. RNA polymerase, again, you can look at it, but Archaea and eukarya have several different kinds. Bacteria, only one, with a single type. We can do methionine. We're going to do introns. Okay. Growth 2, response to antibiotics. Bacteria are inhibited. Archaea, not. And neither are eukarya. So they don't respond to antibiotics, either one of them. Histones we're not going to worry about. Okay, circular chromosome you should know. Bacteria and Archaea both have it, and eukarya does not. And growth at more than 100 degrees Celsius. That's hot, right? Yes, everybody here knows 100 degrees Celsius is hot. Boiling water? Oh, okay. Yeah. Bacteria and eukarya don't grow. Some Archaea can grow at over 100 degrees Celsius. All right, we'll stop there. Okay, have a great... Again, you can ask questions, but try to limit it to the stuff that I'm doing, not extraneous materials. If you have other questions, you can just ask me after class. All right, so I told you to go ahead and take a look at the at the character table. Right? We're at Archaea. All right, so different types. This is a domain, so it's one of the three domains. The Archaea shares traits with both domains, and I did that. I did the tree, the table here. Okay. Most of the organisms in Archaea are extreme. They live in extreme environments. For example, the halophiles. Extreme halophiles live in environments that are very saline or salty. They live in environments that have a lot of saline. Thermophiles are very warm environments, so they're hot. Very high temperatures. So a lot of the Archaea are specialized. They're found in areas that other organisms cannot grow or cannot exist. Another type of Archaea are the methanogens. This refers to methane. So methanogens are strict anaerobes. Anybody remember anaerobic cellular respiration? You can't use oxygen. Yeah, oxygen poisons methanogens. So they do not use oxygen. They use carbon dioxide And what they do is they release methane. Now you find a lot of these in places like swamps. They're what produce that swamp gas. Are you guys having trouble seeing it? That typical swamp gas you hear about, that's what they produce. We're not going to do a whole lot with them. All right, so throughout the semester, you are going to get phylogenetic trees. If they come straight from your book, they're typically showing you exactly how everything common ancestry. But you are also going to get trees that I've designed to help you remember things. Okay? They won't be in your book. They take a long time for me to put together. Okay? The first one you see is in the protease, and we'll get to that in a minute. If you did not get the Elmo portion of my notes for the protease, I printed that out for you, simply because you need that portion of the information. I won't do it for any of the other stuff. Okay? I won't print out any of the others, but just to give you an idea. So this is a phylogenetic tree of the Archaea. Okay? We divide them up in several things. So now notice this is the domain. It includes these three clades. The core archaeotes, the euryarchaeotes, crenarchaeotes, and nanoarchaeotes. Those four. Notice that, is it a monophyletic clade? No, they're not sure. They're not positive the actual common ancestry. So what would you call this? Huh? We had it on the exam. Well, I don't think I asked you actually on the exam. What is it when you don't have a simple dichotomy break-off? You're not sure of the ancestry. Polytomy, yeah. So that's what this is. They're not positive of the exact ancestral break-off or where it occurred. But they have some different types of characteristics. The core archaeotes is a new clade. Not exactly sure where it came. It's just a new clade. The euryarchaeotes, all known methanogens are located in there. So all the methanogens that are known at this point are in that grouping. Some of the halophiles, which are what? They live in extreme saline environments. Okay? Some of the thermophiles are in there. The crenarchaeotes, most of the thermophiles are in there. So you find organisms that live in very hot environments. The nanoarchaeotes are incredibly small. That's what nano means. They're very, very small. And it's relatively a new clade. This is probably the easiest one. That's all the characteristics. I'm not going to give you any ones you have to know. That's what you need to know for the Archaea. Because most prokaryotic cells are in the domain bacteria. And that's what's next. Okay? So this is the domain bacteria. Okay. So I gave you some information. Okay? The vast majority of prokaryotes, and you guys remember what prokaryotes look like. I told you to go back to your book and look at them. So what's the typical characteristic of a prokaryote? No nucleus. Right? They don't have a nucleus. They are a prokaryotic cell. All right? Most of them are in the domain bacteria. Right? Nutritional types. What that means is they get their material, they get their nutrition, their food, in many different ways. And we did that before. Okay, we talked about some of those before. I gave you that table. Yes? Does it sound familiar? Yes, no, maybe so. Oh, good, because you need to know it for the exam. Okay? So you need to know that table, how they get their nutritional material. So that different nutritional types are spread out among the different groupings of bacteria. All right, let's look at the phylogenetic tree. Should have printed this out. All right. So this is a phylogenetic tree. Okay. Again, notice not exactly sure common ancestry. Right, exactly where it divides out. There are several groupings. We have the proteobacteria, the midias, the spirochetes, the cyanobacteria, and the gram-positive bacteria. So do you have to know the terms, those names? Without a doubt. Okay? So you can think, I'm going to put names on one side, characteristics on the other, and say match them. You'll probably have a whole bunch of matching because you're going to have a whole bunch of terms, a whole bunch of names, okay? So yeah, you do have to practice them. So what are some of their characteristics? I wrote them down for you. If you got the Elmo's papers, you've got all of this, all right? The proteobacteria are gram-negative, which means what? The gram stain, you can wash away that gram stain, right? Okay, it doesn't show up. All right. They're very large. They tend to be larger than the others. They're diverse. They can be photoautotrophs. What's that? They use sunlight to produce their own food. Heterotrophs, which means they consume some other organism. Chemoautotrophs, they use chemicals to produce their own food. They're anaerobic or aerobic. So they either don't use food, they don't use oxygen, or they do use oxygen. So those are protobacteria. And there are several different groupings of protobacteria in your book. You have alpha, beta, gamma, delta, and epsilon. Questions so far? Okay.
So, was there alpha, beta, those are kinds?
Those are groupings. Groupings, yeah. Typically phyla or kingdom. Bacteria, they don't separate it out exactly the way they used to. So if you look in your book, they put them out as kingdoms. All right. So look at alpha proteobacteria. Okay? So this is the grouping taken out. There's several of them. Many of the alpha proteobacteria are considered the type of bacteria that were endosymbionts for mitochondria. What they think is, I'll talk about endosymbiosis in just a little bit when we get to proteins, but what they think is that mitochondria actually formed by organisms swallowing, taking in, ingesting, usually through the plasma, so phagocytosis, but bringing this other organism into the body and didn't digest it. It eventually became an organelle, the organelle being the mitochondria. So it's called endosymbiosis, and I'll get to that later.
So it became part of whoever ate it?
it evolved into the organelle known as the mitochondrium because it wasn't digested. And so organisms, what they think is it was organisms that, it was a heterotrophic, polyproteins, heterotrophic cell that took in an alpha proteobacteria, which would have been aerobic, okay, so it probably used oxygen, perhaps. It would have taken that mitochondrium in, but it wasn't able to digest it as food. So over time, it got an advantage, and so produced more offspring, and it was able to replicate. Mitochondria have their own DNA, so they're able to replicate themselves. Okay, so if you look in your book, an example of an alpha proteobacteria, okay, rhizobium. Okay. We'll actually talk about these a little bit more because they're very closely associated to the roots of plants. They're responsible for fixing nitrogen, atmospheric nitrogen. We rely on them. And we rely on them because we cannot fix nitrogen from the air. So what happens, and we'll talk about this actually later, but what happens is they're able to remove nitrogen from the air and put it into a usable form that other organisms can use. So you need to know this one. And that's important, especially since we'll see it again. Archaea is another species. Okay. It forms tumors in plants. So notice that the alpha proteobacteria are not only helpful, they also cause problems. All right, so you really need this one. If I ask anything on that, it will be that, or maybe the picture. Sorry, I thought I was writing on my notes. Okay. Beta protobacteria. An example is Nitrosomonas. This is a soil bacterium. So it's actually a bacterium that lives in the soil. And one of the things that bacteria do is they recycle nutrients. So they're able to decompose, break things down, take nutrients from organic material, put it into inorganic material. It then becomes available for other organisms to use. This is essential. Okay? We use the material that's broken down and put into the soil. Right. Gamma protobacteria. There are different types. There's autotrophic and heterotrophic. The autotrophic that we have here is a sulfur bacteria. It takes energy and it oxidizes hydrogen sulfide. A little bit different than what we're used to. So it is autotrophic. It produces its own food, but it uses a different chemical. Heterotrophic, E. coli. We find these in the intestines of organisms, of animals. And so the E. coli typically are not pathogenic, which means they don't cause disease, but that's not always the case. E. coli can cause serious diseases. It depends on if you get one that is resistant or one that's dangerous. Okay? Do you guys, when you go to another country, so if you're here in one place for a long time and you go to another country, what happens? Yeah, typically you get that upset stomach, and it takes a couple of days for things to work its way out, I guess is a good way. A lot of times what's happening is you're transferring over a different group of bacteria, E. coli. It's the same E. coli species, but not necessarily the same type. Okay? So when you go to a different country, you take a couple of days to do that. All right, Delta protobacteria. I have the myxobacteria here. They produce something called myxospores. These are protective spores. I don't know why, but if you look down at the bottom, the fruiting body, you're going to see this on a lot of them. I got to correct this. But this is actually, these fruiting bodies are actually here. These are fruiting bodies. Okay, so take this and move it up there. Okay. And these fruiting bodies are how they reproduce, so asexual. Some attach to other bacteria. These are actually kind of interesting. So this is a bacterium, and this is Delovigrio, and what it does is it actually bores its way into the other bacterium. And so it drills a hole into the other bacterium to consume them, to feed off them. So go back to that handout, heterotrophic, they consume other organisms. You guys see how I'm doing that? So I'm just working right now, I'm just working on that. That's the only thing we're doing so far. Okay. Are there any questions? Okay. And then epsilon. Okay. Many of these are pathogens, which means they cause diseases. One that's very common is Helicobacter pylori. It causes stomach ulcers. So you remember how it used to be stomach ulcers are only caused by stress? Or is that just because I'm a little bit on the older side? No, I'm not that old. It's been, yeah. No, what they're finding is a lot of stomach ulcers are caused by bacteria. The stress helps to reduce your immune system, and so the bacterium is able to proliferate. But it's caused by bacterium. I'm sure there are other ulcers that are caused by other factors, but mainly bacteria. Okay? And so this is just an example. Again, not the fruiting body. These do not produce fruiting bodies. Okay? So what am I going to ask you on the exam? Because I'm sure right now you're thinking, oh my God, what is she going to ask me on the exam? Right? That's pretty much everybody's... Yeah. No, probably not. Not all of it. You are going to have to know several things. One, you have to know all of these. Okay? You have to know protobacteria. You have to know chlamydias. You have to know spirochetes, cyanobacteria, and gram-positive bacteria. You got to know those. You got to know their characteristics. Right? Without a doubt. Then, some of the ones that I asked. Okay? If I give you an alpha, I might write the species, or I might write just kind of a generalization. Right? You don't have to know all of it. You don't have to identify it. That's also what lab's for. Have you guys been doing it in a lab? Yeah, so you just a little lab. Okay? So that'll be easy. Any questions? All right. Chlamydias. All right. There were two parts that we talked about when we did the wall, right? Do you guys remember that? We said that you could be gram-positive, gram-negative, but what was one of the things that most bacteria had in their wall? Yeah, peptidoglycan. Chlamydias doesn't. It actually lacks it. And it lives in animal cells, which means it's pathogenic, causes diseases. And if you look here, this is in your book so you can see it, that right there is chlamydia. Now, it causes some diseases. One is blindness, but one is non-gonococcal urethritis. Okay? It's a sexually transmitted disease. Basically, it's inflammation of the urethra. Okay? Itis means inflammation. So it's inflammation of the urethra, and it's called non-gonococcal because it's not caused by gonorrhea. So that's chlamydias. Lovely organism. All right, spirochetes. No? Oops. Okay, spirochetes. They're helical, which means they twist. Okay. They're heterotroph. They consume other organisms. Okay. You can find both free-living and pathogenic. Okay, if you look here, two of the spirochetes we have, one causes syphilis, one causes Lyme disease. I'm going to talk about Lyme disease just a little bit. You guys have heard about that, right? Yeah, that's common around here, especially around the mountains here. So what happens are deer get these things, ticks bite the deer, get the material into the tick, they inject it, and you can actually look in your book. It gives you a nice, lovely little picture of Lyme disease. That's why one of the things you do when you go hiking. Have you guys been out in the mountains around here? Now, what's the first thing you do when you're done with your hike? Check, because they like hair. Okay, you know what ticks are attracted to? Yeah. So they'll sit up on a higher area, and they are attracted to sweat. So if you don't sweat, I guess you're a little bit luckier, right? Not really, because if you brush up against them, they're going to get onto you anyway. If you take dogs or animals out there, make sure you check them, because they tend to bring lots of ticks in. Okay? So they'll climb on you, attach, bury into the skin, suck the blood. They're lovely little animals. Okay, cyanobacteria. Oh, I don't know why this is down here. Chlamydia should be up here. When I copied and pasted, some of the things went haywire. Okay, cyanobacteria. If you look here, photoautotrophs, which means they use light to produce their own food. Closely related, right, to other organisms. They have plant-like oxygen photosynthesis. So in other words, they do photosynthesis very much or very similar to plants. Okay. The cyanobacteria tend to produce films. They're marine, so you find them in water. Okay. This is going to be important because we're going to talk about it a little bit. Likely evolution of plant chloroplasts by endosymbiosis. Endosymbiosis, again, is when one organism or one cell consumes another organism. And then that organism, instead of being digested and taken apart as part of the food, which is why they would have consumed it in the first place, it gets incorporated into the cell. And so eventually, over time, it becomes an organelle. It actually works with that cell. All right. Gram-positive bacteria, actinomyces, and mycoplasms. So there are three groupings. We have solitary species, actinomyces, and mycoplasms. Those are three groupings. Some of the solitary, all that means is that they live by themselves. They're not in groupings. And some of them can be pathogenic. Actinomycetes, this is actually used for antibiotics. Okay? So what happens is we can grow these and use the material to produce antibiotics. What I really want to talk about are mycoplasms. Look at these things. These are new. Smallest cells we know. Smallest cells we know are mycoplasms. Not only that, they have no cell wall. Some are pathogenic, which means they can cause disease. Some are free-living. and we find them in the soil. They're really neat. They're relatively new. Okay? They're one of those new cells that's been identified. And they are the smallest that we know of. I'm sorry?
Does that say they're just only found in soil?
No, that's one of the places that you find them is in soil. Yeah, they do some different types of fixing. All right, so those are bacteria. All right, so let's look at different roles of bacteria. Yeah, I'm going to reduce it. Okay, so what do bacteria do? They do what we call chemical recycling. So they actually will take material from things like organic, break them down, and put them back into inorganic material so that other organisms, so they become available to other organisms.
What was the name of that smallest?
Mycoplasm. Right? So they take in material, they can break it down, put it into usable forms, so forms that other organisms can use. They can take material that are in organic beings. Let's say they die. They break them down. They take that material, put it into areas that other living organisms can use. So they recycle. Without bacteria, we would have a serious problem. A lot of material would get left into organic material. When they die, it wouldn't be brought back into the inorganic or into the environment, which means other living things couldn't use it. They would disappear. They'd be gone. So ecologically, they're incredibly important. One of the things we talk about when we come to bacteria, but we'll talk about this more when we do ecology, is symbiosis. Symbiosis is when two organisms live in close contact. So two organisms live in close contact. The larger is typically called the host, and the smaller is typically called the symbiont. So different types of symbiosis. Mutualism, commensalism, and parasitism. I'll have to change that. I don't like that term. Those are the three types. Mutualism. That's when two organisms live together and they both benefit. So they both derive some type of benefit from the relationship. Commensalism. You have two organisms living together, one benefits, one doesn't, one's neither doesn't benefit and it's not harmed either. And parasitism, one benefits, one harms, is harmed. When we're talking about parasitism, we're talking about pathogens. And a pathogen is any organism that causes a disease. Yeah, usually mutualistic and commensalism, since the host is not In mutualism, the host benefits, so it's not going to cause a disease. And in commensalism, the host is neither benefit or harm, so it's not going to cause a disease. So when you talk about parasitism, that's when you're usually talking about pathogen. But it's one that harms. But a pathogen specifically is anything that causes a disease. Bacteria have both exotoxins and endotoxins. Exotoxins are secreted out of the bacterium. So even if you kill the bacterium, it's still there. The toxin's still there. Endotoxins stay inside the cell, the bacterial cell, endo inside. So they stay inside the cell. So in order to release the toxin, you have to kill the bacteria. And what happens is the bacterial cell dies, the cell wall breaks down, and the toxin is released.
So exotoxins, even if you don't kill it, it's still there. I'm sorry, with the exotoxins.
Oh, it doesn't matter if you kill the cell or not, they're still secreting the toxin out. So, different ways to be pathogenic. And then I gave you some things for biotechnology that your book listed. Right? So some of the things that we use bacteria for, experiments. You guys took Bio 6, you know about DNA technology, you did a lot of that in Bio 6. Yeah? I know you did. Okay. One of the things is we're looking at natural plastics. They're actually bacterium that they have found that produce a material that they can extract. How long does it take for plastic to decompose? Just regular plastic. A long time, right? A long, long, long time, right? So they fill up landfills. Well, they think that they can actually, this is one of the things they're working on, they can extract this material and produce a plastic that was biodegradable. That's a big deal. They're not sure they can do it yet completely, but it's a big deal. If you can produce a biodegradable plastic, then it's not going to be filling up landfills, right? So the bacterium doesn't produce this material for us. It actually produces it to store energy, but they've found that they can take this material from these bacteria and produce, perhaps, biodegradable materials, plastics. Let's see. When we're talking about the principal agents of bioremediation, we put a lot of pollutants in the air and in the environment. So what they do is they use some bacteria to remove those pollutants. For example, when we have an oil spill, what do? One of the things they do is they spray it with a bacterium that can decompose oil. Good, bad? It depends on what it does to the environment. Okay, so you can destroy the oil, but it depends on what else it is doing to the environment, whether or not it's good, bad. But it removes pollutants. They use to recover metals from ores. So when you do mining, okay, you can remove metals. Oh, that's an interesting spelling. This should be actually vitamins, not vitamins. Okay. So they produce vitamins. We use them to produce vitamins. Antibiotics, hormones, production of that. Okay, so just some of the things that they have in the book. Bacteria are very important. We use them. All right, that's it. Next.